Phil Hodgkin. I'm from the Walter Rise Hall Institute, and it's my very pleasant duty today to host today's seminar from our speaker, Dr. Chris Jolly. And I'd like to begin by acknowledging the traditional owners of the land in which this event is taking place, the land of the Wurundjeri people of the Kulin Nation, and pay respect to their elders and families past and present. I need to say a few words about the VCCC. If you're unaware, the VCCC is the Victorian Comprehensive Cancer Centre is an alliance of 10 successful Victorian organisations committed to cancer control. Its role is to coordinate the collaboration between member organisations and develop a strategic program of work that integrates research, education and clinical care to maximise patient outcomes in a cost-effective manner. And an important role of VCCC is to organise seminars such as this one. And you can see on the wall there is the upcoming seminar, so if you're interested, please register.
Is that one day or?
Oh, sorry, that's my mistake. I was in a bit of a rush today.
Just add 7 days.
Well spotted.
You're not a doctor.
Uh-oh. Trouble. Okay, so to introduce our speaker today, Dr. Chris Jolly. Chris began his research career at the Macquarie University in CSIRO in New South Wales, cloning genes, plant genes that are involved in defences in wheat. He discovered pura-indolines as members of a large family of lipid-binding antibacterial proteins, and this led to his interest in immunology and a postdoctoral segue at the John Curtin School to study immunology. And that's where I first met Chris in the 1990s. But this interest in immunology escalated rapidly, and he decided to pursue a career in immunology, and he went to do a further postdoctoral stint at the world-famous MRC laboratory in Cambridge, where he worked with Michael Neuberger, a Nobel Prize-winning Cesar Milstein, for a number of years. So, and here, of course, he developed his passion and interest in antibody affinity. We'll hear a lot more about that today. Chris returned to Sydney in 2000 and established an independent NH and MRC funded program at the Centenary Institute, just near at the University of Sydney. He's produced fantastic publications since then in immunity, PLOS biology, PNAS, and I just heard recently in Cell. Just had a manuscript accepted. Congratulations, Chris. Today's presentation, Antibody Hypermutation Evolution on Speed Dial, will tell us more about the fascinating story of antibodies. Of course, during immune responses, B cells make antibodies, and they mutate their immunoglobulin genes at tremendous rates, increasing the affinity of antibody a millionfold over a few days. And they do this by taking advantage of an enzyme called AID. AID introduces mutations deliberately to increase the affinity of antibody, but occasionally that goes wrong and they introduce mutations to things like MYC, leading to some lymphomas. And Chris is going to tell us a lot more about that today. He seeks to understand why DNA repair downstream of AID is error-prone, even oncogenic, and we look forward to hearing more about his recent work and findings. So Chris, over to you. Thank you.
Thank you very much for the invitation to come down from Sydney here today. It's really great, and I've never been in this building before, and I'm really impressed at just walking into it at the integration between the hospital and the research staff, and just what a fantastic place this building is. Now I assume I hit escape to get to my talk. Next. Oh, okay. Next. Oh, on here. Sorry, which one's next? That one.
Yeah, so that one. So just
That one. Or not.
It's just frozen. Right.
There you go. Okay, so I should say I'd forgotten. I'm sorry that I gave Erin a different title for my talk from the one that's on this introductory slide, but it doesn't matter. It's really about the same thing. So what I'm going to talk today about is sort of three aspects. One is what antibody hypermutation is. Secondly, perhaps why you should care about it as a cancer research institute, because it actually does have relevance to B-cell cancers. And then the third aspect will be about how antibody hypermutation might have evolved, and my suspicion that it's actually a very ancient antiviral mechanism that's been hijacked and repurposed to mutate antibody genes. So what is an antibody? I'm sure all of you know that, but I'm going to give you a brief overview of them anyway. We all think of antibodies as these really cool Y-shaped molecules that float around in our bloodstream, and they're able to essentially recognize an almost infinite universe of foreign antigens and produce and bind to those foreign antigens and neutralize them or otherwise clear them from our bodies. So it's a vital part of our adaptive immune system. And antibodies basically, have two ends. If you look at the top half of the Y, those two stems of the Y at the top, they're the sites of the antibody that bind to an antigen, and they're hypervariable. Basically, every single antibody has a different antigen binding site. And then the base, the stem of the Y, is a constant region which interacts with the rest of the immune system and is more or less conserved between all antibodies. But if we look at that variability, hypervariable region in more detail, if you look at this diagram here, the antibody is made up of 4 polypeptide chains, 2 identical heavy chains in the middle, and 2 identical lighter chains on the outside. And we've got the yellow and blue coloring to illustrate that. And if we just look at the variable regions of that heavy chain and light chain, and we sort of plot that variability from left to right on these graphs, then what you see is that the variability is highly concentrated in 3 regions shown in red on that graph in the top left, and the heavy chain. And if you look at the light chain, you see the same thing. There are 3 regions highlighted in red that are hypervariable. And those hypervariable regions, you can see in the figure on the right, form loops at the very outer edge of the antibody. And it's those 3 loops, which are called complementarity determining regions or hypervariable regions, that actually stick onto and bind antigens. And it's the almost infinite variation in those loops that enables antibodies to recognize just about any foreign antigen with very high specificity. And just to illustrate that, we're still using the same color coding system in this particular diagram, where we've just got a fragment of an antibody, which is shown by that circle on the right, with the blue and yellow coloring. And the hypervariable regions that are in red are also in that diagram, but you can't see them because there's a light blue antigen there, which is hen egg lysozyme, the molecule I work with all the time. And the binding of that antibody to that light blue hen egg lysozyme antigen has completely buried all 6 hypervariable regions. You can just see 1 red atom poking out. So all the hypervariable region atoms are completely embedded in that interface between antibody and antigen. So that's essentially how antibodies work. And the reason we can produce literally billions of different antibodies, every one of us is capable of producing roughly 10 to the power of 15 unique antibodies. In theory. In practice, we don't produce that many because we're not big enough. We probably need to be the size of a planet to produce that many. But at any given time, you've got about 1 million unique specificities circulating in the bloodstream. And I'm not going to go into the detail of how all that works, but you obviously don't have 1 million antibody genes in your genome. You only have about 25,000 coding genes in our genome. And the reason we can make all these different antibodies is during B cell development in the bone marrow, our antibody genes are arranged in segments. It's like getting a pack of cards and running it through a bandsaw, and then scattering the fragments of the cards all the way along a piece of string, and then randomly selecting a top section of card, a middle section of card, and the bottom section of card, and sticky taping them back together again. And that makes a new unique antibody gene. And that's essentially how we take a deck of cards of about 50 genes and turn them into 10 to the power of 15 potential genes. But I won't go into the detail of that. That's not what my talk's about. But there's also a second wave of antibody gene diversification. That first wave of antibody gene diversification produces the repertoire that we're essentially born with or develop soon after we're born. But when you're actually undergoing an infection or a vaccination, you have a second round of antibody diversification, which massively improves the affinity of the antibodies you make, so that the ability of those antibodies to neutralize that infection improves about 1 million-fold in a few days. And the purpose of that increase in affinity actually probably isn't to clear that particular infection. If you've got a rapidly growing bacterial infection, making high affinity antibodies in 5 days is too slow. You'll be dead before you've made those high affinity antibodies if you can't already control it with the low affinity antibodies you've got. The purpose of making those high affinity antibodies is to stock bank them for the future, so that when you get a repeat infection, you'll clear that infection incredibly quickly, so quickly you won't even know you had it. Or you will also produce diversity that can deal with related infections you haven't even come across yet. And that increase in affinity is just shown here in the right-hand plot by the increasing steepness of that slope going up. But the other thing that happens is you also change the type of antibodies you make. At the same time as increasing the antibody affinity, you switch from making the starting class of antibody, which is called IgM, to making the antibody which is most abundant in the bloodstream, which is IgG, but also other types of antibodies like IgA, which are secreted onto your mucosal surfaces and onto your skin, and IgE. And these are specialized antibodies that have much more precise targeted specific functions than the starting antibody, which is IgM. IgM is, if you like, a workhorse antibody, jack of all trades, that can function even at low affinity. Whereas these other antibodies, IgG, IgE, and IgA, can really only function if they're high affinity. So how does this change in affinity occur? So it's been known for a long time that this change in affinity is associated with point mutations appearing in those hypervariable regions that I showed you in the graphs before, those red regions. And the number of mutations that accumulate in those hypervariable regions goes on as time goes by with an infection. Antibody genes can accumulate hundreds of mutations if they're extremely mutated. And this whole mutation process goes on in your lymph nodes. So when you get an infection in your lymph nodes, typically your throat lymph nodes, if it's a respiratory infection, will swell up. And part of the reason for that is that those lymph nodes are filled up with B cells, which are proliferating like mad and mutating their antibody genes at the same time. And so what's going on in that lymph node is essentially Darwinian evolution on speed. That the B cells are proliferating very rapidly, about once every 6 hours. They're mutating their antibody genes with each round of proliferation, and then they're competing with each other to see who makes the best antibody. And he who makes the best antibody wins and goes on to do another round of proliferation and mutation, and he who makes the worst antibody drops dead. And so it's just Darwinian evolution occurring in the lymph node. And you can see that in this section of the lymph node. That's not my section, by the way, it's a colleague's, and I've forgotten to attribute it because I've actually forgotten who it is. But the regions where this hypermutation is going on are shown in blue, the GL7 regions of that lymph node section. So antibody mutation is induced by a specific enzyme called AID, activation-induced deaminase, which was discovered in 2000. And what activation-induced deaminase does is really what the rest of my talk is going to be about, but it targets very specific regions of the antibody genes, which are shown by these red curves, which basically shows how the AID targets the 5' ends of genes, and then it's targeting tails off as you move away from the 5' end of the gene. And the attack by on antibody genes by AID both introduces point mutations, but it can also introduce breaks into the DNA that make the DNA recombine. And that's how AID causes antibodies to change isotype, which I'll go into more detail. So look, I'm just going to skip that slide, we don't need it. From a cancer point of view, the reason a cancer audience might need to care about AID is that AID is essential for effective antibody-mediated immunity, and without it, you're immune deficient and you will get really nasty viral infections. But the downside of AID is that AID, if you have chronic infections that go on for months or years, especially if they go on for years, then AID will keep on mutating antibody genes, and that's fine. But like all things in biology, the targeting of AID for antibody genes is not perfect. About 1% of AID mutation is in off-target genes that AID shouldn't mutate. And those off-target genes are all genes that are highly transcribed in cells that are actively proliferating. And many of the genes that are highly transcribed in cells that are actively proliferating are cancer-causing genes called oncogenes. The classic one is MYC. So what happens is that AID can target antibody genes, as you can see on the left here, but on the right, at the same time, at a low level, it could be targeting an oncogene like MYC. And if it happens to introduce a break into MYC as a result of that targeting and a break into the antibody gene at the same time, when the B cell goes to do class switching, recombining antibody genes, it can accidentally recombine with the oncogene. And what that results in is the oncogene is now being transcribed in the incredibly powerful antibody gene promoter, which is one of the most powerful promoters in a B cell. And that means that that oncogene is permanently turned on in that B cell. And that can lead to B cell cancer, such as follicular lymphoma, Burkitt's lymphoma in Africa, and multiple myeloma. And the really best evidence for this is a theory that's been in place for many years, and the absolute concrete evidence that that is actually what happens came out in Cell a few years ago, where Michelle Nissenbach's group in New York showed that chronic plasmodium infection in humans in Africa is the driver. It drives genomic instability in B cells and AID-dependent African Burkitt's lymphoma, which is different from the Burkitt's lymphoma we get in the West. So it's a plasmodium infection, which is malaria, which actually causes the Burkitt's lymphomas via off-target mutation by AID. So what does AID do? So I mentioned it's activation-induced deaminase. So what that means is that AID targets one of the 4 bases in DNA. DNA's got 4 bases, A, C, G, and T. C is cytidine. And AID deaminates cytidine, which means it hydrolyzes off an amine group and turns that cytidine into a uracil. So C to U mutation. A U in DNA is essentially the same as a T. So what that's doing is it's turning a C into a T, which changes the information that gene codes for. The other thing it does, in turning a C into a U doesn't base pair with G. So the AID-induced mutation is doing 2 things. It's changing what that base codes for. It's also creating a mispaired base in the DNA, a little bulge in the DNA due to a mispair. And so I'm going to refer to that as the founder mutation that AID causes, C to U. And that's all AID does. It doesn't do anything else. It just does that one job. And that was published in Nature by my former mentor in 2002. So what happens if you've got a U in the DNA? Well, usually U actually occurs in DNA spontaneously at a low rate all the time. You're getting U's in your DNA right now. You get about 100 per cell per day, just spontaneously. And that is in fact why we have T in DNA and not U. In RNA, we use U. We have A, C, G, and U in RNA. But in DNA, we have A, C, G, and T. And that is because C can spontaneously turn into U in DNA anyway. In RNA, that won't happen. So by having T in DNA, our cells have evolved, all life on Earth has evolved to be able to distinguish between uracil in DNA as opposed to thymidine in DNA and know that they're 2 different things. And that's really critical for the integrity of our genomes. So what can happen? So there are DNA repair processes in place that look for U's and get rid of them. But if the DNA repair processes don't see those U's and that cell just undergoes cell division and duplicates the DNA, then what will happen is that one daughter cell will inherit that mutation, the U, and put an A opposite it. And now you've got a permanent CG to TA mutation, essentially. And one daughter cell would be unmutated. And that happens actually quite a lot in B cells, as it turns out. But what is more likely to happen, and in fact it happens about 2/3 of the time, is that there's a specialized enzyme called UNG2, and its job is to look for uracils and get rid of them. So it goes through the DNA looking for uracils, and when it finds one, it flips the uracil out of the base pairing and chops it off, but it leaves the DNA backbone intact. And that's the process called base excision repair. And there's actually a whole bunch of base excision repair enzymes that all do that job, but they all recognize specific types of DNA mutations. So UNG recognizes U, and that's all it recognizes. So I show that when the uracil is being excised, but the DNA backbone's intact, I show that line with the red dot. So that's what's called an AP site, an apyrimidinic site, AP site. And we know that UNG is involved in antibody mutations downstream of AID, and I'll go through that in a minute. Now, what would normally happen is that when UNG excises a uracil like that, there's a dedicated DNA repair pathway called base excision repair, which I show circling back. And it uses a series of steps, which I won't go into in detail, but uses a series of steps to completely excise that damaged base and then replace it with a C, and then ligate the DNA back together, and you get back to where you started from with an intact piece of DNA with the correct DNA sequence in it. But the critical thing to notice about that repair process is it involves cleaving the DNA backbone. The enzyme APE, apyrimidinic endonuclease, so it's an endonuclease, it introduces a nick into the DNA. And that's really critical, that point. So that nicking is how AID actually causes DNA breaks. AID itself doesn't cause DNA breaks. It's the DNA repair enzymes downstream of AID, specifically APE1, especially APE1, that causes those breaks. And on the right, you can see a whole bunch of DNA repair pathways that all feed into that same base excision repair DNA repair pathway, which I won't go into in detail. So that would restore the correct base. And in fact, that goes on. Most DNA repair induced by AID is in fact the correct DNA repair, putting the C back in place. So most of the mutations AID introduces actually just get repaired and turn back to where you started from. But what seems to happen in B cells is that UNG can act to remove that uracil, and for reasons that my research is zeroed in on, but until I did that research, we didn't really know why. For some reason, the whole repair process gets interrupted halfway in B cells, and the repair process doesn't complete, and the B cell replicates that DNA before base excision repair is completed. And the theory has always been that if it replicates before that nick is introduced, then when the DNA polymerase that's synthesizing new DNA comes along, it'll encounter that A basic site and it won't know what to do. And it'll just put any base in there, which I've shown as an X. And we know that you have to have special kinds of enzymes called translesion DNA polymerases to cause antibody mutation. And translesion DNA polymerases especially evolved to deal with sites like A basic sites, AP sites. So they're capable of replicating DNA across opposite the AP site, but they'll put any old nonsense in there. So we know that the formation of certain types of mutations at GC base pairs require both AID and UNG. And if you don't have UNG, you don't get those mutations that I'm showing on the right there. And those mutations specifically are transversion mutations at CG base pairs, which are the worst kind of mutations. Transversion mutations are the mutations that are the most likely to cause an amino acid change in a coding sequence. But they're good for antibody mutation. These mutations are good for antibodies because it makes them more diverse. But the uracil can also be processed by another DNA repair pathway called mismatch repair. So all these DNA repair pathways I'm talking about, their job is actually to make the DNA, to fix the DNA, not to mutate it. Their job is to fix the DNA, not mutate it. But mismatch repair can also recognize the U, not specifically by recognizing the U, but by recognizing that the U and the G are mismatched. And if mismatch repair is recruited to this UG mismatch, then it will also cause a break in the DNA, but it will also excise a long stretch of DNA, about 200 base pairs long, that includes the U. And that way it gets rid of the U, and then DNA repair enzymes are recruited to put new DNA back in place. Again, for unknown reasons, unusual DNA repair enzymes are recruited in AID-induced mismatch repair, and they generally repair the original mutation introduced by AID, but at the same time, they put in new mutations at nearby AT base pairs. And so that ends up giving you permanent mutations in antibody genes at AT base pairs. So by having a combination of AID, error-prone repair via UNG, and error-prone repair via mismatch repair, you are able to get any kind of all possible types of point mutations that are introduced into antibody genes, and are also introduced into any of the bystander genes that AID might accidentally mutate, such as MYC. So this was all nutted out by Michael Neuberger in the early to mid 2000s, and that pathway is now essentially accepted as being the correct pathway. I should say, sorry, that as I mentioned earlier, that because AID recruits UNG and base excision repair, the base excision repair pathway can introduce nicks in the DNA where the uracils were. So if AID introduces uracils on both DNA strands, which it does, then if you've got nicks on both DNA strands downstream of AID, those nicks end up forming double-stranded breaks. And double-stranded breaks are particularly genotoxic, and they're what cause oncogenic translocations, but they also cause class switching. So it's the double-stranded breaks primarily that are the oncogenic aspect of AID's activity. But the fundamental question, ever since this model was developed in the early 2000s, the fundamental question I seem to have, which for some reason other people didn't seem to have, I don't know why is this process error-prone? It's all very well mapping out that this is how the mutations occur, but why are the mutations occurring? Because these DNA repair pathways we're talking about in every other cell are very high fidelity. They repair errors, they don't create errors. So why are they so error-prone in these cells? And I had a suspicion that this all had something to do, there's a note in the bottom there about telomere loss, which I won't go into. AID can also attack telomeres on the ends of DNA, but I won't talk about that today. So my research over the last 15 years has been driven in trying to understand why AID causes error-prone repair. Because I think if we know why it causes error-prone repair, then maybe we've got a chance of stopping some of these oncogenic downstream effects of AID. And so to answer those questions, I just want to point out to you that there's this protein called UGI showing, I keep pointing at things you can't see. There's a protein called UGI shown in red there. And it's a bacterial virus protein, which is actually capable of blocking specifically the activity of UNG. So I'm going to use that as a tool in the slides I'm going to show you next. So I had a suspicion that the cell cycle was the key to why AID is so mutagenic. The cell cycle is a series of steps that every cell has to move through in order to prepare to replicate its DNA, to actually replicate its DNA, to divide that DNA up between two daughter cells and then actually divide. That's called the cell cycle. And DNA repair is intimately linked with the cell cycle. because you don't want to progress through the cell cycle unless all the DNA repair steps have been carried out in the correct sequence. And UNG, this enzyme that acts downstream of ARD to be so mutagenic, canonically, it's particularly active in S phase. Its job is to look for uracils in S phase and get rid of them. And I had a suspicion that maybe UNG was acting in a different phase of the cell cycle in hypermutating B cells. And just to illustrate why I thought that, it was because B cells proliferate extremely rapidly in vivo, much more rapidly than normal cells do. I'm not going to go into what this data is, but this particular graph you can see on the right, which is still unpublished data, what we're able to determine with that graph is that B cells divide every 6.2 hours when they're proliferating in the germinal center, which is phenomenally fast. And so it doesn't leave the cells much time to carry out DNA repair, which can be quite a slow process. So this is an illustration of the cell cycle. It's got three main phases: G1, which is when cells prepare to divide; S phase, when they actually synthesize the new DNA, and that's usually quite a long phase; and then G2 phase, when they check that completely synthesized DNA hasn't got any errors in it, they fill in any other gaps, and then they move into mitosis, which I haven't shown you, which is when they divide. And canonically, UNG is predominantly active in early S phase. But it is also active at low levels in G1 phase. And it's long been hypothesized that the reason for that is in G1 phase, viruses will quite often replicate in G1 phase as a way of escaping a lot of the mechanisms that cells use in the nucleus to prevent viral replication. And so UNG's G1 phase activity has long been thought to perhaps have some antiviral activity, which I'll go into more later. So what's our tool for looking at all this? We've got this protein called UGI, which is shown here in blue, which binds to the enzymatic site of UNG and blocks that activity. So we reasoned that if we could express UGI in B cells in a cell cycle regulated manner, then we could ask, when exactly does UNG act in the cell cycle to mutate antibody genes downstream of ARD? So what we did was we came up with this concept of sequences called degrons, which are about 100 amino acids long, and they come from naturally cell cycle regulated genes. And what these degrons do, I've shown the degrons in this diagram in blue or red. What degrons do is that they recruit any protein that they're fused to will be very rapidly degraded when the cell moves into a particular phase of the cell cycle. They recruit, I won't go into detail of how they do that, it works through polyubiquitination. But these degron domains, any protein that carries these degron domains will only accumulate in a very specific phase of the cell cycle and will be degraded in the other phase of the cell cycle, even if the gene encoding that protein is constitutively expressed. So you can see that in figure B here, where this is a FACS plot showing DNA content from left to right. So cells that are in G1 phase have 2N copies of their DNA, and cells that have completed DNA replication have 4N copies of their DNA. And cells that are in S phase actively synthesizing their DNA are anywhere in between. And on the vertical axis is the expression of a fluorescent protein that's fused to the degron. And what you can see is that when a blue degron is fused to a protein, we've got GFP or MCO2, GFP is green, MCO2 is orange. When you've got a blue degron fused to that protein is only expressed in G1 phase, and in every other phase of the cell cycle is completely degraded. And there's an opposite degron, which we call psych, which is shown in red here. And when a fluorescent protein is fused to that degron, that fluorescent protein accumulates only in S, G2, and M phases and is absent from G1 phase cells. So by fusing proteins to these degrons, my lab was able to very precisely control the expression of restrict the expression of any protein we liked to very specific phases of the cell cycle. And this movie here just illustrates that. So the bad microscopy is a movie from my lab, because my lab are not good microscopists. And here we have an orange protein fused to one degron and a green protein fused to another degron. And you could see that the two colors are mutually exclusive. So as the cell, that's not going to play again. As the cell, so the cells here in S phase, it's synthesizing its DNA, it splits, it loses the green protein, and then it starts to accumulate the orange one. There's other people who also use degrons for other purposes at the same time as us, and I've stolen their movie. And they did it much better. They make much better microscope slides than I do. And you can see the degrons switching from orange to green in that movie. Now, those aren't actually the same degrons as the degrons my lab produced, because we invented them independently, but they do the same thing. So What are we going to do with these? So we have a system for looking at antibody mutation in mice. You can't, there's no in vitro system for looking at antibody mutation. We have to do it in vivo, so we do it in mice. And we have, I won't go into details of it, but we have immunoglobulin transgenic mice where every B cell responds to a particular antigen, a synthetic antigen called Hennig lysosome. And when we take those switch Hel B cells that react to Hennig lysosome and adoptively transfer them into immunized mice, the B cells from those immunoglobulin transgenic mice will dominate the B cell response, and virtually all of the antibody produced in the host mouse will come from those genetically engineered B cells. So what we do is we take those genetically engineered mouse B cells, we put viruses into them, retroviruses into them that express our cell cycle control protein of interest, purify the cells that we've successfully got virus into, and then adoptively transfer those cells into a new mouse, which we then immunize with the antigen that we work with. Let them sit there and participate in a normal immune response over a week. Then we can sort those cells back out, one cell per well, and then sequence the antibody gene present in that cell. It's a transgenic antibody gene, so we know exactly what its sequence is. And we just sequence one cell at a time in a 96-well plate, and we can very accurately and very precisely then map the mutations that have been introduced into usually about 50 cells per experiment. And we can accumulate very large amounts of data very quickly this way. So I won't go into how we do those experiments in detail, but so we put this protein that blocks the activity of the UNG enzyme, and we expressed it either in G1 phase or only in S phase using our dead ones. And then we accumulated mutation databases using those recombinant proteins, when those recombinant proteins are being expressed in the B cells. And I'm just showing this as the sort of data we get. We're looking at the antibody gene from the 5' end to the 3' end, and you can see a skyline of mutations where each column is one base in that DNA sequence and the number of mutations that have accumulated at that base. And I won't go into detail about what those mutations are, except to say, to try and make this data visually easier to understand, all the mutations that occur above the line are essentially mutations that AID alone would introduce. Whereas all the mutations below the line, mirror image below the line, are mutations that require DNA repair enzymes downstream of AID to be introduced. So you can see in the top graph where we've got rid, it's a UNG knockout mouse, and it's also a mismatch repair knockout mouse. So it's UNG minus M minus. So it doesn't have any of the enzymes that will process AID-induced mutations downstream of AID. So all you get is AID-induced mutations. All the mutations are above the line, and there aren't any below the line. Well, there's a couple, but essentially there are no mutations below the line. Whereas we go down, and then we've got single knockouts of base excision repair or a single knockout of mismatch repair. And then at the bottom, we've got a wild type mouse. And you see in the wild type mouse in the bottom that there's roughly equal numbers of mutations below the line and above the line, because about half the mutations are introduced by AID alone, and the other half of the mutations are introduced by the DNA repair processes downstream. So when we do that sort of analysis and use our inhibitory protein that would cell cycle restrict to either G1 or S phase, what we find using that is that all the mutations that are introduced by UNG downstream of AID, they all depend on UNG activity in G1 phase and only in G1 phase. And they, oh, sorry, I've got ahead of myself. So the purpose of this experiment is to ask, sorry, I've got ahead of myself, is to ask, so UNG introduces a particular type of mutation downstream of AID, but when does it do it? Look, actually, I won't go through the raw data because it's really quite complicated to go through, and I will just give you the conclusion. But basically, if we block UNG activity constitutively in the cell cycle, we get rid of all the mutations that UNG can introduce. And that's what the black columns are. If we block UNG activity just in G1 phase and leave S phase UNG activity intact, we get the same result. We still block all of the UNG-induced mutations, and we've got two different degrons giving us the same result. But if we block UNG activity only in S phase, we have very little impact on UNG-induced mutations. So the S phase UNG activity is not relevant to antibody mutation. And just to show that that's not an artifact, the final column on the right shows if we mutate that degron so it no longer gives cell cycle regulation, we go back to completely blocking UNG-induced mutations again. So the short story there is that the UNG activity in G1 phase and only in G1 phase is critical to antibody-induced, to AID-induced antibody mutation. And it turns out when we look at class switching, this process of switching from the starting isotype of antibody IgM to the downstream isotypes, IgG, it's the same result. And again, this is a very complex diagram with a lot of detail, and I'll just tell you the take-home message, which is that it's G1 phase UNG activity, which is critical for antibody class switching, and S phase UNG activity is essentially irrelevant. And the figure on the right just shows that our constructs actually are blocking UNG activity. So the take-home message from that is it's G1 phase UNG activity that's critical to antibody mutation. But the canonical activity of UNG in protecting the genome, the housekeeping DNA repair, is predominantly in S phase. So the antibody mutating activity of UNG is quite distinct in the cell cycle from its normal DNA repair role. And so this is, I think, our first major clue as to why AID is so mutagenic. It's inducing DNA repair pathways in phases of the cell cycle when they're not supposed to be active, or when they're not supposed to repair DNA at least. And that, we published that in JXMed a few years ago, 2012. There was always a niggling doubt in the back of my mind that we might be wrong, because there's lots of complications in those experiments. But it was very nice to see last year that two groups basically used our ideas and did follow-up experiments using essentially the same ideas. And what those groups showed was that they cell cycle regulated AID using exactly the same tools that we'd invented, and they got exactly the same result, that if you overexpress AID in an AID knockout mouse, you have to express AID in G1 phase to get antibody mutation, and S phase AID doesn't do anything at all. And another group at the same time in the same issue of JXMed showed that what actually happens with AID in vivo is that AID gets pumped in and out of the nucleus. And in actual fact, AID is only present in the nucleus in G1 phase, and it's excluded from the nucleus in all other phases of the cell cycle. So that, I think, quite definitively showed that the basic scission repair, DNA repair downstream of AID, is restricted to G1 phase, and it's mutagenic because it's acting in G1 phase. And I'll go into why it's mutagenic in G1 phase later. But what about the other DNA repair pathway, mismatch repair? This is the other pathway that processes AID-induced mutations. We weren't, unfortunately, able to cell cycle regulate mismatch repair enzymes in the same way that we were able to cell cycle regulate phase decision repair. We tried, but it failed. But it turns out we've been able to come up with other ways of answering this question anyway. So again, canonical housekeeping mismatch repair acts exclusively in S phase. And mismatch repair's job is basically to clean up all the nucleotides that occasionally get misincorporated by DNA polymerases. When DNA polymerases replicate DNA in S phase, like everything in biology, they're not perfect, and every now and then they introduce the wrong base. The mismatch repair's job is to get rid of those bases, and it does it in S phase, and it does it by making these large excision patches essentially, and then inducing resynthesis across those excision patches to fix up the errors that have been introduced. And the key thing about S phase mismatch repair is that because of the structure of the replication fork, the mismatch repair complex, which is huge, it knows which strand is the original DNA template that presumably doesn't have mutations in it, and it knows which strand is the newly synthesized DNA strand. And it will always excise a newly synthesized DNA strand, knowing that it's getting rid of the newly introduced mutations. Now, that particular canonical mismatch repair pathway uses a bunch of very well-known enzymes, but which I won't go into. But AID-induced mismatch repair doesn't use the normal enzymes. The DNA polymerase that is recruited in canonical mismatch repair is called Pol delta and Pol eta, but the DNA polymerase that's recruited by AID-induced mismatch repair is an error-prone polymerase called Pol eta. And there's also a ubiquitination step that is involved in AID-induced mismatch repair of the DNA sliding plant. There's a molecule called PCNA, which you can think of as a donut. And it's PCNA that actually slides along the DNA, and the DNA polymerase is attached to the PCNA and slide along the DNA as passengers on PCNA. So if modifications, ubiquitinations or other modifications occur to PCNA, then that alters the speed of the replication fork, and it alters what DNA polymerases will be recruited to the replication fork. So AID-induced mismatch repair involves a non-canonical polymerase, Pol eta, that's what that Greek character is, and it also involves a modified form of PCNA. Now, both of these things have been known for a number of years, but up until recently, it remained a complete mystery as to why these particular enzymes involved in AID-induced mutation. So we had a suspicion, which has culminated in the publication of PNAS just this year, that the reason that those weird enzymes are introduced are involved in AID-induced mismatch repair is again because of the cell cycle. That if AID is inducing mismatch repair in G1 phase, then all sorts of things are going to be very poorly set up for mismatch repair. One of the key things about the cell cycle is that the supply of DNA building blocks, nucleotides, is heavily restricted by the cell cycle. So even in S phase, we only have enough nucleotides in our nucleus to synthesize about 100 kilobases of DNA at any given moment. But the genome is 3 billion base pairs long. So there's a continuous real-time supply of nucleotides to replication force as the genome is replicated. In G1 phase, there are virtually no nucleotides available at all. And the reason that those nucleotides are so heavily cell cycle regulated, and that's what those graphs show, the reason they're so tightly cell cycle regulated is that it's an antiviral mechanism. Viruses want nucleotides too. Viruses want to replicate. We are all riddled with viruses, and these viruses want to replicate in our cells a lot, and we don't want them to. So one of the ways we stop them from replicating is by undersupplying DNTPs, and especially in G1 phase, we starve them of DNTPs. It's very difficult for a virus to replicate in G1 phase. And HIV gets around this by destroying one of the key enzymes involved in this whole nucleotide supply chain, in restricting nucleotide supply. So nucleotide supply, like all things in biology, has two sides to it. You can synthesize nucleotides, and there's a key enzyme called ribonucleotide reductase, which is a rate-limiting factor for DNTP synthesis, and that's a very tightly cell cycle regulated enzyme. There's a more newly discovered, and it's been known about for decades, there's a more newly discovered enzyme called SAMHD1. And SAMHD1 destroys DNTP, so it's the yin to RNA's yang. And SAMHD1 is also cell cycle regulated in the opposite way to ribonucleotide reductase. So in G1 phase, SAMHD1 is happily destroying nucleotides, depriving viruses like HIV of DNTPs. But in S phase, SAMHD1 activity is quite low, and ribonucleotase activity goes up, and nucleotide supply goes up. What HIV does is it produces a protein that destroys SAMHD1. That's one of the ways HIV gets into macrophages. So I reason if AID is inducing mismatch repair in G1 phase, then the DNA repair pathways downstream of AID are also being starved of nucleotides. And mismatch repair is a DNA repair process that involves the excision and resynthesis of really quite long tracts of DNA, at least 200 base pairs long and quite possibly a lot longer. So in S phase, which is the high DNTP example in this diagram, that's all fine. Mismatch repair can excise huge chunks of DNA, and then they can be resynthesized and everything will be fine. The canonical PCNA, which is shown in yellow, and the canonical pol delta shown in pink, will be able to happily go along and synthesize new DNA with high accuracy and repair the mutations. If you try and do the same process in G1 phase when the DNTP supply is very low, then the pol delta, the canonical pol delta, is really going to struggle to synthesize DNA. It's going to stall. And that stalling will induce modifications in PCNA, the introduction of enzymes, the recruitment of enzymes that are more tolerant to this low nucleotide environment. And then you synthesize the DNA now with these error-prone enzymes that are nonetheless tolerant of a low nucleotide environment. But a side effect of that is that you're going to introduce random mutations due to the error-prone nature of that enzyme. So that was the theory. And we proposed that theory in 2010. So how do we test it? So we decided to use our Degron approach, our cell cycle approach, to cell cycle regulate nucleotide supply. And basically did two things. One was we used our viruses to overexpress one of the subunits of ribonucleotide reductase that's very tightly cell cycle regulated. That subunit is called R2. So you can see in the right-hand side at the top that R1, the R1 subunit is pretty much constitutively expressed through the cell cycle, but the R2 subunit is detectable in SG2&M, but pretty much absent in G1 phase. But if we overexpress R2 with a virus, then now we can get R2 to be, it's still cell cycle regulated, but we now get R2 to be present in G1 phase and present in huge amounts in S phase. So we use this virus that overexpresses the R2 subunit to look, oh, and we did another thing. So we also made a knockout mouse of SAMHD1. So SAMHD1 is the enzyme that degrades the NTPs in collaboration with Rob Brink at the Mega facility in Sydney. We used CRISPR technology to make a SAMHD1 knockout mouse, and we actually got 3 independent lines and just worked with 2 of them, but all 4 of them, actually produced 4 independent alleles, and all of them knocked out SAMHD1, but we just worked with 2 of them. So we have a SAMHD1 knockout mouse, and we have a way of overexpressing R2 using a retrovirus. And what happened with the SAMHD1 knockout mouse, as we'd expect, just look at the graphs on the G0 phase graph on the left is the best, but the other phases, the other graphs mimic it, is essentially the SAMHD1 knockout mice had a 40-fold increase in DNTPs, and that's what you'd expect. If you get rid of the enzyme that's degrading DNTPs, the NTPs can now build up to much higher levels than they would normally occur, especially in G0 phase and in G1 phase. Interestingly enough, though, we haven't collected much data on this, but when we overexpress R2, we didn't cause any detectable changes in the NTPs, which was a bit of a surprise, and that's what the black bars shown there are. Nonetheless, overexpressing R2 did make the cells more susceptible to viral infection. You can see a twofold increase in viral infection on the left there in cells that are expressing R2 versus control cells. And that hints that even though we couldn't detect a change in nucleotides with our relatively crude nucleotide measurement tools, when we did a biological assay, a viral transduction assay, we could see that the R2 expressing cells were actually more sensitive to viral infection because the virus has a better chance of getting to the nucleotides. And you can see on the right that, as we'd expect in a SAMHD1 knockout mouse, their cells were also more susceptible to viral infection, which is already well established and expected. And that's because of the changes in nucleotide levels. So we predicted that a loss of SAMHD1 or a gain of R2 would selectively inhibit mutations that depend on mismatch repair, because we are predicting that those mutations depend on nucleotide supply. That was our prediction. And to cut a long story short, so we used this adoptive transfer model again to do exactly the same experiments as before. Lots of transduction, lots of FACS sorting, lots of DNA sequencing. And we get these mutation skylines. And what you can see straight away if you look at these mutation skylines is the mutations that depend on mismatch repair are shown below the line because their mutations occur downstream of AID, and they're shown in green. They're predominantly at A or T bases, whereas AID alone just causes mutations at G and C bases. And U and G predominantly causes mutations at U and G, at C and G bases as well. So you'll see in the wild type mouse at the top that you've got lots of black and blue mutations above the line due to AID. You've got lots of green mutations below the line due to mismatch repair. And you've also, although they're hard to see, you've got black and blue mutations below the line due to mismatch as well. But when we knock out SAMHD1, you can see that all of those green mutations below the line, most of them disappear. Not all of them, but most of them. And you can also see, that's the bottom graph, sorry. In the middle graph, where we overexpress R2 with a virus, we get essentially the same result. That the green mutations below the line more or less disappear. Not completely, but largely disappear. There's also, however, some subtle changes in the black and blue mutations below the line, which weren't expected. So in detail, the whole bunch of bar graphs here, but it's probably easier if I just tell you what happens, is that overexpressing R2 does indeed inhibit mismatch repair-induced mutations at A and T base pairs, and it pretty much doesn't do anything else. And that's what we predicted, which was nice. But SAMHD1 knockout was a bit more complicated. It actually didn't specifically inhibit mutations at A and T base pairs. It pretty much blocked any transversion mutation, whether it was at AT base pairs or at GC base pairs. And transversion mutations are a particular class of mutation which hadn't really been, mutations at AT base pairs had never really been divided in any meaningful way into transition and transversion mutations before, because there was no evidence that they occurred by any separate mechanism. But it turns out that it looks like they do. But transversion mutations are the most mutagenic mutations. They're the ones that are most likely to cause a detrimental change in an antibody, in an oncogene, or a useful change in an antibody gene. So one thing that we wondered was that were our treatments simply inhibiting mismatch repair or base decision repair by unexpected means, as opposed to just influencing nucleotide supply as we expected, because of these unexpected changes in transversion mutations. And look, I'm not going to go into how this particular graph tells us that it's not, because it would take half an hour to explain what these graphs represent. So you just have to take my word for it that we are very confident that our SAMHD1 knockout and our ribonucleotase overexpression are not inhibiting mismatch repair or base decision repair. And the reviewers of our paper in PNAS accepted that we proved that with these graphs, so I won't go through it because it would take forever. So why we were publishing it, so that half of that story is now published. The SAMHD1 story was published in Proceedings of the National Academy of Science this year, but we haven't been able to publish the ribonucleotide reductase data yet because it didn't cause any changes in measurable changes in nucleotides, and the reviewers of the paper were very bothered by that. So we've kept that data back until we can sort it out. And what that led to was me going through the literature trying to find out explanations for what was going on with ribonucleotide reductase. And it turned out there was a paper 8 years ago that showed that ribonucleotide reductase is actually actively recruited to DNA breaks in G1 phase cells, and that recruitment of ribonucleotide reductase to G1 phase breaks is critical for the repair of those breaks, but it's not critical for repair of DNA breaks in S phase. And it's thought that what's going on is that when you're repairing those G1 phase breaks, you need some local synthesis of DNTP, so the locally acting enzymes have all the building materials they need to synthesize new DNA and repair it. And in S phase, you don't need that local recruitment because ribonucleotide reductase is already constitutively active and there's an ample supply of DNTP. So that's the theory. So I thought, well, maybe what's going on in our cells is that when we overexpress ribonucleotide reductase, we're able to get a local increase in DNTPs at the site of AID-induced mutation sites, but we don't get a global increase that we can actually measure. We can't measure local concentration of DNTPs. There's just no tool to do that at the moment. We can only measure them globally. So we wondered, does AID induce local recruitment of ribonucleotide reductase to DNA repair sites? And to test that, we made a bait protein. So this bait protein is able to block the recruitment of AID to, block the recruitment of ribonucleotide reductase to G1 phase DNA repair sites. It blocks them. It blocks that recruitment blindly. It doesn't matter what kind of DNA repair it is. This bait protein will block the recruitment of ribonucleotide reductase. And what it does is it basically blocks the binding site that would normally recruit ribonucleotide reductase to DNA repair sites. And we found that when we, this is very preliminary data, but when we overexpress this bait protein in our adoptive transfer system, we've only done this twice so far, we essentially blocked all of the mutation that is induced by mismatch repair downstream of AID. So that theory looks like it might be correct. And interestingly enough, overexpressing SAM-HT1, which completely obliterates nucleotides in a G1 phase cell to an even lower level than normally present, also blocked pretty much all mutation downstream of AID. AID-induced mutations themselves were fine, but all the mutations that are introduced downstream of AID, like DNA repair enzymes, were blocked if you overexpress SAM-HT1. So I think what that's saying is that all the DNA, this is preliminary data, but I think what it's saying is that all the DNA repair pathways downstream of AID require a nucleotide supply. And what's going on is that those DNA repair pathways, part of the reason they're mutagenic is that there are nucleotides available. There's enough nucleotides available to get some DNA repair going, but there aren't enough nucleotides available to get good DNA repair going. So it's really quite a simple concept at the end of the day. And if you make that situation even worse by expressing even more SAM-HT1, you just don't get the DNA repair at all. It's complete, a bricklayer can't build a brick wall without bricks, and we're essentially depriving the bricky of any bricks. So that's quite a simple concept, but it's not the whole story. And look, I think I won't give what the rest. There's another aspect to why I think AID causes mutations. There's a second explanation on top of that nucleotide supply thing, but I don't think I've got time to go through it, unfortunately. But it's being published in PNAS this year anyway, so if you're really interested in it, you can read about it. But I will finish with this about this whole talk. So why are these mutagenic DNA repair pathways existing? If G1 phase mismatch repair is so mutagenic, if G1 phase UNG basic repair is so mutagenic, why do they exist in our cells at all? Why on earth, after billions of years or millions of years of evolution, do we have highly mutagenic DNA repair pathways active in our B cells? And it doesn't look like they're just active in B cells, they're almost certainly active in other cells as well. What on earth is the point of that? And I think the point of that is that they're actually antiviral. That these G1 phase DNA repair, its job isn't to repair DNA at all. Its job is to stuff up DNA. And specifically, its job is to stuff up viral DNA. And the reason I'm thinking along those lines is illustrated in this picture I stole from a recent review, or not that recent, but reasonably recent review. And there's an innately antiviral enzyme called ApoBec3G, and it's very closely related to AID. AID is actually a part of a family of enzymes which are called ApoBecs. And AID was actually the first one to where its function was understood, and then all these other relatives that were called ApoBecs, it's now realized what their function is. They're also cytidine deaminases. And ApoBec3G is potently antiviral, especially against HIV. Again, HIV crops up again. ApoBec3G destroys the HIV genome, and patients who are mutated for ApoBec3G are highly susceptible to HIV infection. But HIV, being a very clever virus, produces a protein called VIF, which blocks ApoBec3G activity. So it produces VIF to block ApoBec3G activity, and it produces another protein VPX, I believe it is, which is the one that targets SAMHD1. It has a whole bunch of little tiny proteins that just target, block antiviral proteins. And so APOBEC3G deaminates the HIV genome. HIV blocks that, but it doesn't block it completely. And then the downstream DNA repair, its job is to destroy the HIV genome downstream of APOBEC3G. And UNG is one of the enzymes that's involved in that downstream destruction of HIV genomes. But also SAMHD1 is involved in that downstream destruction of the HIV genome as well, by preventing HIV from replicating in the first place. So you can see all these DNA repair enzymes are cropping up in now emerging antiviral pathways, and they're also reappearing in my favorite pathway, which is the antibody mutation pathway. So I think the antibody mutation pathway is simply a mutagenic DNA repair pathway that originally evolved to attack viruses. And then at some point in the development of the adaptive immune system, our ancestors said, well, okay, let's take those pathways and attack our own antibody genes with them to make our antibody genes mutate to be more effective against viruses. So it's using the same tools to do a slightly different job to get the same outcome, which is antiviral activity. So I think, I can't prove this theory as yet, but that's my sort of grand theory at the moment. This acknowledges all the people over many years in my lab who have done all this work. The whole model was first developed by Adam Cook, who has now unfortunately left science to work for the TGA. A very talented PhD student, George Charbin, has gone to have a very successful career at University of New South Wales. Daniel Bosniak, who's now a medical student, collaboration with Robert Brink, and my most recent PhD student, Eddie Tiento-Sapol, who has just submitted his PhD thesis, and various people who've worked in my lab as research assistants and funding from the HMRC, Cancer Council and various other bodies. And one thing I finally want to finish with is a piece of work that, it's one slide, a piece of work that I was not the driver for, but was a major collaborator on, which has just been accepted for publication in Cell. So any of you who might be involved in the HMRC grant review or know people who are, tell them that we've just got a paper coming out in Cell. And basically what this does is there's been this mysterious death through kidney disease of immunocompetent mice all over the world, especially in Australia. And we've discovered using viral metagenomics a new virus, which we've called mouse kidney parvovirus, which causes kidney failure in immunocompromised mice. This is the graphical abstract that we had to produce for Cell. I won't go through all this, but it's a new virus called mouse kidney parvovirus. Why would you care about that? Well, if you work with mice, you need to care about it. But on top of that, we've also discovered through data mining that there's a monkey equivalent to this virus. This is a very divergent virus. It's very different to any other known mouse parvoviruses, which don't cause disease, I might add, whereas this one does. But there's a monkey, quite closely related monkey virus, which has just been partially sequenced from macaque poo. And I think it's pretty fair that there'll be a human. If there's a macaque one, there's probably a human one. So I think somewhere out there, this virus might be causing disease, kidney disease in humans, which is what we're hoping to embark on to translate this work into something useful to patients. And thank you very much. I'll stop there.
Thank you very much, Chris. Fantastic tour of antibody mutation. We invite some questions from the floor. While we're getting organised, let me ask you about, you said that AID was excluded from the nucleus during
this
phase?
Yeah.
So that's why it wouldn't be invoking mutations. But what about UNG? You said that's actually expressed in S phase.
Yeah,
so why aren't you repairing the S phase?
I think, well, yeah. Okay, yes. My answer to that is UNG is very low abundance. It's actually, if you do a Western blot of UNG, you'll see nothing. And it's not because the antibody is no good. It really isn't very much of it. And that's why we haven't done many experiments over expressing UNG, because it's a bit non-physiological. That's why we use the inhibitor protein instead. So I think what's going on, UNG's job in S phase is to actually track with replication forks. And it just goes along with them. It's actually bound to them. And it's part of the whole replication complex, if you like. So its job is to look for uracils in real time as the DNA is being synthesized and get rid of them before they get replicated. And of course, because there's only a very limited amount of UNG in the cell, and there are thousands of replication forks going at a given time in this phase, each replication fork is only about 100 kilobases apart. So I think basically it's all taken up with being at replication forks. And the actual antibody genes are only present in a replication fork for a finite time. So I think there is a little bit of repair that UNG does in AID-induced repair that UNG does in S phase when UNG happens to be there. but the rest of the time it's just not there. It's been titrated away to the rest of the genome. Whereas in G1 phase, it's just gagging about, waiting for something to happen, so it can wander around aimlessly.
Any other questions, please?
You can hardly imagine that this enzyme, AID, is of real advantage to human. It must have been introduced by retrovirus, surely, because there that mutation makes sense.
AID is definitely advantageous for humans, don't worry. Your immune system is far more important than getting cancer. You know, for most of human evolution, you know, we lived to 45 years of age, and as long as we got, you know, as long as a few grandparents survived to pass on the, you know, our social evolution, your role in producing children was over with at 40. You know, you only needed a small number of people to become wise to keep the, you know, the collective knowledge going. The rest of us were expendable. So you're not going to get cancer before you're 40, but you're going to get a lot of infections. So it was much more important to fight back. You know, the rate of bacterial infection in hunter-gatherer communities is vastly higher now. Like, we don't need our spleens, but hunter-gatherers need their spleens. If they have a traumatic accident, they're going to need their spleen. We don't need ours at all, because we're just too clean, basically. So, you know, the adaptive immune system is absolutely critical to surviving childhood and surviving young adulthood, surviving injury, wounding, etc. Cancer just isn't an issue in pre-agricultural human society, or even pre-industrial human society, I would say.
So we relied on the repair mechanism not being perfect to introduce that new system.
Exactly, yeah. It's fuzzy logic, if you like. It's deliberately not, you know. Yeah, I mean, I think that's how evolution works. Evolution works by things not being perfect, because that makes them adaptable. Perfect tools are not particularly flexible.
Any other questions? Yes.
Again, like a sort of side question, you mentioned about the rapidity of the replication
Of the B cells, yeah.
Was that 6 hours around the replicating infection, or is that?
That's in my adoptive transfer model, so it's not an actual infection. And it has the other caveat that there's a trick we use in our adoptive transfers. We use sheep red blood cells as an adjuvant. And there's something a bit weird about sheep red blood cells. I think they're essentially recruiting memory T cells even in a naive mouse. So I'm not sure that a primary response necessarily involves B cell, a true primary response. I could do this, but I just don't have fun with it at the moment. That particular set of data was a lot of work just to get that graph. But I think it's unlikely to be different. I think 6 hours is probably right. Yeah, I think I actually got that data ages ago and never got around to finishing to publish it. But I don't think it's what that data shows that other people's data may not show. And I think you showed quite a lot in your in vitro system. You got quite long S phases. We didn't. In that system, which is still unpublished, I worked out S phase, but I can't be absolutely short in mass of being 89 minutes. But there's some provisos on that because the data could be contaminated with cells that aren't cycling at all. So I can't be sure, but I think yes. So I won't hold my hand to my heart and swear on it. But based on that data we have at the moment, S phase is 89 minutes, which is phenomenally fast.
So taking a broad correlation with that, then you mentioned the connection, the model of infection. Do you find is there any kind of corollary with people with B cell lymphoma who had, you mentioned the infection and the is it numerous infections?
No, it doesn't. Having lots, yeah. So I don't want to make people think that being immunized is a bad thing or anything like that. No, because they're all short burst things that give you high affinity and then shuts down and stops. So don't think vaccination is going to give you cancer. I must absolutely state that vaccination is not going to give you cancer through this pathway. Absolutely not. It's got to be chronic and it's got to be the same infection. So it's got to be the same B cells. They're being activated over, just endlessly activated. And it's just a numbers game. If you fire a shotgun at a target often enough, you know how it's going to go out to the side. That's just all it is.
Does this mouse virus only appear in lab mice?
No, it's in the wild as well, it turns out. It's not going to infect humans. Quite confident it doesn't infect humans because our animal handlers have been handling these mice for decades unknowing. And mouse fanciers for hundreds of years have been their lives would have